welcome to the channel, or if you're already a subscriber, welcome back. Today's notes video is going to be on mutations. And no, unfortunately, I'm not talking about the mutants that you see with the X-Men. These mutations that I'm talking about are changes in the DNA sequence. Mutations can be brought about through several ways. You could have a mistake that occurs during DNA replication or in the process of transcription. That's one way to get a mutation. Another way is that you may inherit a mutation, meaning if your mom or dad had a mistake in DNA replication or transcription and it's passed to you, you may have a mutation that way. So I'll write in parentheses here that it's passed genetically. And there are certain things in our environment that can promote a mutation in our genetic code. When I talk about environmental, I'm referring to one of three primary causes. You might be exposed to toxins, which could alter the structure of your DNA, cause a change in the sequence. You could also be exposed to UV radiation. That's how we get skin cancer. We have too much exposure to the UV rays of the sun, and it changes the DNA of our skin cells and causes our skin cells to replicate uncontrollably, and that's how you get skin cancer. Exposure to certain chemicals can cause mutations in your DNA sequence. Okay, so these are the reasons for a mutation and what a mutation is. But now we need to talk about all the different types of mutations that are possible. To do this, I'm going to make a concept map, and I'll start with mutations in the middle. So that's our focus. Now, for mutations, we can have two primary types. The first is called a point or substitution mutation. You could really see it written either way as a point or substitution. So it's a good practice to have both kind of in your head. The other type of mutation is called a frameshift mutation. So those are your two types. A point or substitution mutation is a change in one nucleotide. So those are often easier to pinpoint. A frameshift mutation could get complicated because it causes a change in the polypeptide. So frameshift mutations can really mess you up. We have two types of frameshift mutations. One is called an insertion, where we quite literally will insert an additional nucleotide into our sequence. The other type is going to be deletion. We take one nucleotide out of the genetic sequence. For point or substitution mutations, which can sometimes be called non-frameshift mutations, there's three types. The first is nonsense. That means that the one nucleotide change just makes a stop codon come up so quickly when it's not supposed to, and translation is going to stop too soon. Could also have something called missense, where a change in a nucleotide or a substitution of a nucleotide results in a different amino acid being added to the chain. And last, you have a silent mutation. Those would be when you have a change of a nucleotide, but when you use the codon chart, it still gives you the same amino acid